Thank you again for the invitation and the very kind introduction. So I needed to actually, similar to Steve, I needed to mention I'm still at venture like trained as background. I cannot really claim myself as computational biologist. I can say I actually interact with computational biologists a lot. So today I actually want to share both wet bench, also dry bench tools developed in the past pretty much 10 years time in my group. So basically, again, as in the central dogma, nowadays we have a really beautiful chromatin architecture through Hi-C, and the textbook is starting changing it. And because AlphaFold, you have a really complicated structure like a protein. All these AlphaFold have generate a lot of a beautiful 3D structure. The question for a very long time is really messenger RNA has actually remained as a single line in the textbook. And it's actually not really the case if in the past 40 years, anyone drop any kind of RNA with different sequence content, you actually can see it will form these things which we call stem loop, GU base, AU base, also GC base. Also, a lot of study in the nucleic acid chemistry area, you actually can find a more complicated structure like RNA triplex, pseudonote, like rubber switch type, also G-quadruplex. So why it's happening, textbook still remain as a single line, that simple structure as for RNA is majority time is all these studies focused in the in vitro condition. So you don't know whether or not it's truly folded in vivo. So of course you want to be able to know their biological function. So this is really actually global like an effort across in the world from a lot of group. About 10 years ago, we're starting to develop this kind of a chemical probing-based RNA structure profile method, which basically is using kind of two type of chemical. One is a dimethyl sulfate. It's very small. It can penetrate into different type of species, also different subcellular compartment. And what it do is we'll target to the base, Watson Crick phase base of nucleotide base when it's single-stranded. So you don't form AU base or CG base. They will attach this dimethyl sulfate, will attach methyl group at the Watson Crick phase. Then when you're actually doing reverse transcription, because you block the Watson Crick phase, the position, so then reverse transcriptase will stalling at these nucleotide which got a modification. And if you have a lot of stalling of the nucleotide position actually at this specific nucleotide, then this nucleotide tended to be modified by chemical. In the other way is this nucleotide tended to be more single-stranded. So we generate a deep sequencing library, capture all these kind of stalling information during the reverse transcription. Then through like bioinformatics tools, we can develop something called chemical reactivity. You have a more stalling at a specific nucleotide, then you have a high chemical reactivity. And there's another chemical called a shape compound, which actually, rather than targeted to the nucleotide base, it's isolated 2' hydroxyl group when the RNA is single-stranded. So you basically, similarly, you make a block on the RNA, then you can stop reverse transcriptase. Then through years, a couple of recent development is now you're probing DMS in a certain condition, you can go to 4 base the same time. So DMS also can target the AUCG as the same as shape. And also another recent development is when you do reverse transcription for 3 hours rather than 1 hour, you can actually introduce mutation at this modified position. So again, through deep sequencing, you can measure the mutation of a certain nucleotide level. There you can decide whether or not it's actually a high chemical reactivity as a more single-strandedness. So again, when I started my lab, I was really ambitious. I looked into textbook. You see pretty much from the beginning of a transcribed RNA towards processing, then translation, degradation, also, microRNA target. It looks in the textbook, everything is single-stranded. So it's like a simple line as RNA. So that time we thought, okay, we maybe can actually try to, update what we know on the textbook. So what are the model organism we're using is Arauda cediana. Also, through years in the past, we're working on almost all these different type of plant species. And as a Miles just actually talk about, plant habitat in 20-ish degree, it actually can give a relatively thermostable RNA structure. So it kind of become a really good system to understanding structure functionality. So I won't go detail, but I will point to some part of this study. So in the past 10 years, we have a slightly understanding about the pre-mRNA do have a certain level of structure. We found like a 2 nucleotide upstream 5' splice site control splicing selection. Also, we found these single-strandedness in the pass site, which can be additional annotation for the pass site. We also actually find, secondary structure control translation. Also, these very special G-quadruplex tertiary structure, which I'm going to talk in detail, which actually suppress translation. Also, actually, secondary structure, also these G-quadruplex structure inference degradation. I won't go detail, but if you are interested, we also study microRNA target. We find the structures do have a really strong impact towards microRNA target efficiency. Also, another part of maybe some people interested in protein phase separation. RNA itself can also form phase separation. It actually can drive in itself to actually form RNA-driven phase separation. So just to really focus on very new method that we recently developed. So RNA secondary structure, I think I talked 2-3 years ago when in the similar II workshop, but it's actually a lot of a method that has been developed. But today I'm going to talk about these new methods which can measure the in vivo folding for this beautiful tertiary structure called the RNA G-quadruplex. So RNA G-quadruplex is formed by a G-rich sequence. So what it do is you're using both Wilson-Crick phase, also Hugenstein phase. So then you have a 4G formed together using both Wilson-Crick phase, also Hugenstein phase form these hydrogen bond. So then you form these 4G plane structure. So when you have 2 layers of this 4G plane structure, then you actually can form these G-quadruplex tower structure, more tertiary structure. So from the sequence, you can see you have a 2 GG, then loop sequence, then GG, loop sequence, then GG, loop sequence, then GG. You can have a 2 layer, you also can have a 3 layer, 4 layers, 5 layers. Okay, so if you have a potassium, sometimes sodium nitrate, these ion can actually stabilize this kind of structure, make it more stable. Also, like a lithium can actually destabilize because it's too small, it couldn't holding this tertiary structure. So then, again, this is actually discovery like 30 years ago, and everyone thinks it should fold in vivo, right? But then there's a lot of tools try to develop, try to understanding whether or not it's fold. Of course, still, a lot of effort still don't know whether or not it's fold. So it's really accidentally through our chemical probing method, we certainly notice we maybe actually can detect its folding status in the living condition. Again, if you still remember this shape chemical, I told you it's acylated 2' hydroxyl group when the RNA is single-stranded, right? So very interestingly is if you fold the G-quadruplex as a folded status, these last G of every G repeat is exposed their 2' hydroxyl group more preferentially than other nucleotide. So that means this last G of every G repeat is tended to react with this chemical more preferentially. Okay, again, because it's react more preferentially, you will have these kind of a specific stalling pattern at this last G of every G repeat. So through deep sequencing, you can measure this kind of a stalling pattern. This is really uneven distributed. Again, if you folded this G-quadruplex G-rich sequence into a lithium condition, which you won't be able to form G-quadruplex, you lost this preferentially modification. So what you see is more even kind of a distribution of your stalling pattern through the deep sequencing. So when we observe this kind of a pattern of a modification by this shape, we thought maybe we can actually set up a transcriptome-wide in vitro folded standard and also in vitro unfolded standard. Then we basically compare with in vivo, right? So then we took a biophysical analysis, the folded G-quadruplex as this example. When we do the sequencing for that, you can see in the lithium, this G-quadruplex tend to have a more even distribution of a stalling pattern. Then when you actually have a potassium folded, you actually more skewed towards these G is more preferentially modified and actually stalling at this position. So then we think, okay, maybe we can actually now expand into genome-wide. So then we did a genome library just to actually compare in vivo to the in vitro folded status and the in vitro unfolded status, generate something called a folding score. So again, just the example I show you, so the lithium you see more even distribution, potassium is more uneven distribution, then the in vivo is much more similar to the potassium condition. So you know this G-quadruplex in the cell, it's actually folded. So then how about the transcriptome-wide? We actually see a global majority, most G-quadruplex in Rhabdomyceliana is actually folded towards a folded status close to like a 0.75. So majority of them is actually under folded status. And then if you check rice, which is a high GC content genome, you see much more stronger folding and more like G4 is folded through a whole transcriptome. So if you remember 2016, David Patel using another method called DMS-based method, tried to say in the human you don't have, in the mouse you don't have these G-quadruplex folded. So we were wondering why in the plant we can detect so many G4, but in the mouse you couldn't find it. Not surprisingly, is actually it's all the stress response when in the plant is associated to that. So my postdoc think, okay, maybe because it's semi-stable in the plant, that's why we can detect so many G4 in the living cell. So then we actually focus on a very interesting function as a response to code. So then my postdoc did is actually threw the plant into 4 degrees for 2 hours. Then probing in vivo, probing again, just to try to compare folded and unfolded status to see what happened. Interestingly, we can find at a 22 degree, you can see this G4 is more similar to the lithium condition like unfolded status. But where you put into 4 degree, it's similar to the potassium folded status. So that means just G4 under 4 degree, only for 2 hours, it can trigger G4 formation. So you can actually see these G-quadruplex can form where you actually treat it with code. So where you actually see globally changing when you put the whole distribution C there, when put into 4 degree for 2 hours, almost all these G4 either tend to be stronger or you have a more G4 actually form. So in global wise, you can see these code can actually trigger G-quadruplex formation or enhance the folding. So then when we saw that, my postdoc said, okay, what code is really due to these G4, why these G4 is triggered, what are their molecular function, right? So the first thing we did is, translation regulation. We try to see whether or not the typical G4 is inhibited translation. Do we see that? But because it's maybe 2 hours of code, we couldn't see very obvious difference. So then my postdoc started to measure the stability of RNA. So we did a transcription arrest assay to actually tracking down the stability of that. Take every message shorter, so we basically can detect those code-induced G-quadruplex happening in the 3-UTR, these genes. In the 22 degree, it has relatively faster degradation. But where you move to code, it becomes quite stable. So it seems like when in the code, in the 3-UTR, you actually trigger these G4 formation, you will actually get this RNA more stable. So then we did a transient assay with, 3-UTR with Y-type G-quadruplex, these code-induced G-quadruplex, and we actually mutated a couple of G to A, abortion the G-quadruplex folding capability. What you can see, 22 degree don't have G4 folding, so you don't see difference. But in the code, you see when you abortion G4, you actually can see its degradation faster. Okay, so as a plant biologist, we want to see whether or not we can have a strong genetic impact, right? You think about it, it's non-coding G-quadruplex structure, right? So we knock out this plant mutant, so you don't have this gene at all in this plant. So when in the normal temperature, you don't see difference, but in the cold, you can see this knockout gene, you see longer root than this cold suppress plant root growth phenotype. So normally, if you put a plant in the cold, you will actually have a shorter root. But because you don't have this gene at all, it's completely a knockout, it's insensitive to cold. When you complement the whole gene with this 3-UTR had a cold-induced G-quadruplex, complement to the mutant, you can restore this kind of a cold suppression on the root development case. But if you actually just complement with the 3-UTR, changing this G4, G to A, then you see it's similar to the knockout mutant, because at 4 degree, this RNA going through faster degradation, then it's similar to your knockout mutant. So we kind of can see a little bit of genetic impact. So then we think, okay, maybe there's actually selection there. So we did actually 1,000 plant transcriptome try to measure G4 density. So what we can find is G quadruplex is highly enriched in those plant transcriptome where this plant is habitat in more cold area. So we can find a significant correlation between temperature and G quadruplex density in the both 5-UTR, 3-UTR. So we think these G quadruplex can be a potentially stress sensor. Interestingly, is if you still remember in the mouse, people claim there's no G4. So that's why in the human condition, normal condition, you don't see G4. But if you apply the stress, you see similar things. You see G quadruplex formation where it forms. It's in the 3-UTR. What it do is actually it's try to stabilize the RNA in response to stress. So this is a co-submission we actually did with Pavi's group when we actually accidentally found out we actually coordinate the discovery at the same time. So we actually built up this G4 atlas, which you can search any kind of species, which available data set. Everyone can looking for your favorite gene when another G4 is formed. I don't go details, but we are also interested in another type of tertiary structure called I motif, which is C-rich motif, which are also very interesting towards its architecture, also structure-wise. So we develop a lot of method towards prediction. You can check this is a website. It's also based on a machine learning method. We develop it's automated machine learning, so you can update all the time as well. So the other technology I really want to focus is, as everyone was talking about, RNA structure is super dynamics. This is one single sequence, how many possible conformation in the in silico prediction in via the fold. Okay, so just the one sequence, right? So the problem is for years in the whole area, people want to know is how to obtain individual RNA conformation in the living condition. So whether or not we can detect the in vivo. So we really actually started by demonstration a new methodology in this very famous plant long non-coding RNA called QL. Give you an idea, so this FLC is a gene which is important when your plant facing vandalization, so a very long treatment of cold. So this gene will be actually suppressed during the cold, and then the plant will starting flowering. Okay, so how it suppresses this gene is through its antisense non-coding RNA called QL. This QL will actually associate a chromatin of FLC, then suppress expression of FLC. Then you got a flowering. Okay, so when I joined the center 10 years ago, I have a very famous collaborator, Yiliang Ding, is working on this whole thing. She said, can you probe a QL? If you notice, if it's a luminous sequence, it's really tricky because QL had a different isoform. So I don't know which reads align to which isoform. So we specifically develop a new method for probing this QL. So again, when we have a chemical plant, you treat it with a chemical. So all the RNA single stranded will be modified by your chemical. So then you can have all the isoform there. Each isoform will have a different RNA population with chemical modification. Remember, I told you we can actually introduce mutation during reverse transcription. So you generate cDNA with a mutation profile. Then you generate double-strand DNA. We can actually then attach to the double-strand DNA to PacBi nanopore now. So you basically can actually generate a single molecule reads. So each hi-fi reads contain mutation information of chemical modification. Each reads can derive one structure conformation. So now if you're sequencing 1 million reads, you're gonna end up 1 million structure conformation. Then we can cluster similarity between the structure, then generate these structure clustering. So this is one of the major QL isoforms. So after the sequencing, we can actually obtain millions of reads of different structure conformation. Then once you cluster, you can find 3 major cluster of structure. So again, because it's code-related long non-coding RNA, so you kind of treat it with code, right? So in the code, 2 scenarios are happening. One is actually certain structure conformation going down, certain conformation going up. Of course, you also can detect a very code-specific conformation change. So you can actually identify these both conformational changing, also proportion change. When you have these high resolution of a structure, that's the moment you can find the interesting site which associated with these conformation changes. We find a highly dynamic region, then we design like a 4 mutant, generates a much more accessible conformation of a QL. So once you actually have a more accessible, like an open kind of structure of a QL, that means it can associate a chromatin with FLC, the sense gene chromatin, much more stronger. When you associate a chromatin much more stronger, you suppress the FLC expression much more stronger. So then you have early flowering. So we can actually tuning these kind of really kind of a high detector resolution functional structure motif. You can actually really impact their function. Okay, so again, during the COVID, I end up a lot of big data of RNA structome. So we have a lot of sequencing data. So I was really worried how we possibly transcriptome-wide to find, structure, functional structure motif, which allow us, to editing to actually have, transgenic line can actually see the impact of RNA structure. So then we're talking to a very AI expert called Dr. Curley from University of Exeter. So during COVID, we actually teach each other kind of a large language model. So this is basically is a foundation model currently we're using for the ChatGPT as a draft email, data analysis, also reading paper. So I don't think I need a lot of introduction. So basically, difference between machine learning, deep learning is you're basically training a baby. This is I got an explanation from the AI expert. It's basically traditional machine learning, deep learning is like you're training a baby with 1,000 Shakespeare poems, and you expect the baby to actually really come up with a poem in Shakespeare style, right? So the foundation model or large language model, you basically just train the baby as a PhD in literature with almost all the knowledge, grammar, everything. Then you give her like 1,000 Shakespeare poems, then they can actually really write a poem in Shakespeare style. So that time we actually discussed whether or not we can build something like RNA ChatGPT. So then we're starting these long-term training step. So again, what we really want to do is, if you can actually got all these human language with actually typical phrase, also like a word, how we possibly from the nucleotide sequence we know, okay, this is FLC gene. Of course, that's gonna take a longer time. So the first step we move to is actually generate these plant RNA foundation model. So basically, we're using, as I said, we have these 1,000 plant transcriptome sequencing data. We thought, okay, how about we just do large language model on these plant transcriptome. So what are we actually using for training? We're using RNA sequencing, RNA annotation, so where it's 5-UTR CDS, 3-UTR region, and we're also using in silico RNA structure prediction. We're using two kind of standard. One is a 13 nucleotide, all the possible conformation, because we want to give more information rather than one specific structure as the lowest minimum free energy. So we have 1,000 assemble possible structure conformation of 13 nucleotide. Also, we have 100 nucleotide window size. So we try to increase, maximize the training capacity. So what we did is put all these information to these transformer platform, which is about a 12 layer, which we pretty much go through can get pretty much only not the data high. Its data time is 52 million parameters, so it's quite good. Then we have a better one now. So basically, is we try to extract all these information like self-training. So give a little bit actually measurement, which we normally, it's actually more better towards to computational people, is one, we actually using two other kind of species, which is not trained. We send their transcriptome into our foundation model. Then we ask, okay, can you actually predict 5-UTR, 3-UTR CDS correctly? So we basically can get a really high performance, higher than popular method like ESM2 or BERT method. Because you think about it, we train on the plant-specific transcriptome, so that's why it learned the plant species annotation far more better. So this is what I say. It really depends what are things you want to learn. Then your learning library is really important. So then we also did these typical things as a fine-tuning. So what we did is we take a 5-UTR over 10-20,000 genes across in Arabidopsis and the rice, two species, and they are corresponding translation efficiency data. So previously, when we try to learn any pattern, we only know a correlation, or basically it's just so hard because 5-UTR is about 300 nucleotide. If you do massive mutation, that's 4 to the 300 times data point. It's just not really possible. And now we don't need to do so many experiments. We just take a 5 UTR over like 10,000, 20,000 gene and their corresponding TE. Now we convert it into this foundation model, then generate the learning. We try to actually come up with a background subtraction. We either shuffling the sequence of 5 UTR or shuffling the label. So we actually shuffling the translation efficiency, then running through the model again. Again, this model is actually running more like a 50% of random kind of learning. So then we generate something called attention contrast matrix. So basically it's subtract what we learn from the correct true label and subtract the random one. So then we generate these attention contrast matrix. So the first thing we actually did is we average all the attention metrics crossing all these 10, 20,000 genes of their 5 UTR. What the interesting things come up is no matter it's a sequence or structure motif, apparently when it's close to the AUG, it tends to be more important to the translation efficiency, okay? So not a surprise, in this area, we can find a Kozak sequence. So I needed to say I was surprised because I know during model training, I never tell the model there's something called a Kozak sequence. So this is something which, you know, the model it can generate itself. So then we try to find out whether or not there's any structure motif. So if you remember, people always talking about, you know, there's a GC percentage correlated to something or certain strong structure, or but exactly on your gene, which part of the motif is important to your translation is really something we want to get, right? So what we did is we actually have these RNA structure predicted seed library. Then we link this seed library with attention contrast on every single gene 5 UTR. Then we basically just very simple. If attention higher, we corresponding to the base pairing region. Then align with all the structure motif, then see which one is actually coming out. So then we actually can separate a cluster doing simple clustering with high translation, low translation. Then interestingly, we can find all these motifs associated with low translation is these kind of a 4 base of a CG pair. So again, as I said previously, everyone knows CG higher, it's actually correlated to translation. Now we can really identify what kind of a CG motif really associated with low translation. So interesting, we also find that these high translation motif. You think about it, your 5 UTR won't be completely single-stranded because it will actually introduce endonuclease cleavage, right? So that means it will form certain level of structure. So this is what we got. It's a relatively weak base pairing, AGCU repeat. And surprisingly, in one of the literature in Drosophila, they found actually AGCU repeat sequence was corresponding to one case of a high translation event. So now we kind of understanding, okay, you do need a certain structure actually maybe is actually promote translation. So when we know exactly the gene, exactly the motif position, we can make a mutation. We can changing low motif to high translation motif. You see translation enhancement. You also can actually changing high translation motif to low translation motif. There you see the suppression. Okay, again, for years, people was talking about a G-quadruplex is regulator translation, inhibit translation, right? So here, because we have these measurements in this specific condition of the plant, you actually can identify exactly 21, confidently identify these 21 low translation associated RNA G-quadruplex motif. Again, when you make a mutation, basically just changing G to A, you can actually abortion increase translation. So you actually don't have a translation inhibition. So during that time, my postdoc comes through. Here's its AI model, right? Can we actually do the prediction? We can actually tune in the translation as the level we wanted, rather than we just mutated or actually just disrupted. So then we think, okay, we go back to this trace, right? So you have the attention averager, which closing to the AUG, you have more impact to translation. So my postdoc said, okay, if G-quadruplex is suppressed to the translation, so can we actually, when simulate to move it out towards 5 UTR? So what happened? So the model tell you when you move out this G-quadruplex towards 5 UTR, the beginning of 5 UTR, you see it lost the importance. So that means it maybe don't really matter to the translation. Okay, so this is a simulated one, right? So then we actually really run the experimental, which is the most shocking part. So this is a native G quadruplex. We identify suppressed translation. When we move close to AUG, even pretty much close by to the AUG, you see translation suppression. When you actually move out, it's enhanced. If you move out towards the beginning of a 5 UTR, you completely lost the inhibition of a translation. So that's the moment I was like really convinced, this whole learning is doing something, So just to give you an idea is now we actually have a lot of all these examples which we kind of starting learning how to design the RNA, and rather than originally people using MIMI to enrich motif, we now give additional information where you're gonna put your motif, then it will have its function, right? So why call foundation model? Because now we're starting training a lot of things, RNA degradation, RNA binding protein, RNA modification, everything now starting going through that. We think we can actually try to identify every part of a sequence of a gene, whether they actually have a different functional output. And we also can possibly identify these sequences which can have these trade-off factor, which multiple function linked to one kind of a sequence or motif. So I just tried to make a little bit of attention as a collaboration with Steve West, also Stuart Wilson. We actually generated recently a much more powerful animal model, also training on pretty much 1,000 animal species with both sequence, annotation, also all the RNA structure. Differently, this time we go for a mixture of expert structure, which is a DeepSeq kind of a type of a training. So this is much more powerful. It's give you pretty much 577 million parameters behind this model. So far more better performance and more powerful towards this language model. I think everyone was actually talking about why you don't using the whole NCBI. Actually, at the very beginning, we did using NCBI. But the problem is that if you notice, NCBI had 64% of a bacteria genome. So if you want to find a translational regulation element, you end up biased towards SD sequence. So that's why we separate different species to try to understanding a lot of things which may be more eukaryote specific or certain plant or animal kingdom specific. So we also actually generate a plant DNA foundation model, which now we can actually learn a lot of cis elements, functional elements on the DNA as well. So just it's actually gonna free open all these models very soon. The RNA foundation model is there. So if anyone want to use it, all the resources is open. Yeah, so the last I just want to acknowledge my group and all the collaborators. We actually really collaborate with everyone. All the wet bench method, we actually now, if anyone want to probe your genes, it's just like 1 to 2 weeks on the nanopore. Super easy. It's not that difficult any longer. So we try to make all these wet bench tools easier. Also, all the computational resources is online. So let me know if you want to, working on specific question or these things, we try to help. Yeah, thank you very much.
Great, thank you so much. Any questions? Maybe I'll start. So G quadruplex definitely affect, but do you know how? Do you have like an insight into what protein binds to it, or?
So firstly, if you're talking about the moving part with actually control of translation, we think is because when you're closer to the ATG, your ribosome density is higher. So the helicases don't have enough space to coming down to unwind it. But if you move outside a little bit, you don't compete with ribosome towards the helices coming down. So this is what we think we're testing now. So whether or not it's due to the density of ribosome actually association causing the helicases don't have chance.
So you're talking about a structure that masks something. So
I think a structure will be there. Then you have a certain, so it will actually, you don't have enough time inside the cell, the helicase is sufficient enough to unwind it, then you actually can turn on the translation. But it's in the 5 UTR, your ribosome is not stuck in there or actually occupant there. So your helicases have enough time to actually unwind it. So that's why we think this is the reason why it's actually have this distance effect. So it's still under testing.
Okay, so, but when you started, you talked about how RNA is folding and unfolding to different structures, but is G quadruplex the same, or is it just
it's
folding
the same. So the reason why we think plant is a good system, first, plant had a lot of G2 layer, so it's much more flexible. So when it's actually folded, unfolded, it's relatively can be regulatory. The reason why in the human a lot of people was argument, but I think it's still the case, is if you have a 3-layer, so the kinetics of forming G4 is slower, but once it's formed, it's very stable. So that means it's harder to unwind it. But if you have a gene, it's always have a a block of G4 there. So that means your translation, degradation, everything is stopped, right? So you won't be able to create these kind of strong structure. So that's why we think is in the cell is tended to be more flexible G4 rather than very strong block on the RNA. So this is what we think as a genome wide.
Okay, super interesting. Yes.
I think the previous panel discussion, there was this question like, what is the ground truth? And people frequently say X-ray structure or EM would be the ground truth. But as here we can see the structure of the RNA molecular changes under different conditions, and that's like important for understanding the biology. So I don't know if you could give some advice for the machine learning people here, like what would be a good tool for predicting RNA structure? Like what would be important.
So I needed to say 2 parts. So first, I'm still RNA structure people. I still needed to say we should create AlphaFold RNA in the way. But as I said, the foundation model had another side product is actually can predict RNA structure very well. But it depends what is your benchmark, right? So I think a current stage is make a secondary structure prediction far more better is the key, because that bring the next level of accuracy of a 3D structure. And truly is, I think, a ribosome RNA, rubber switch, also, tRNA had a very kind of a stable structure. But the messenger RNA, personally, I think it actually requires a lot of dynamics because, a lot of regulatory, also RNA binding protein is actually interact heavily. So I think you need a certain dynamic there. But whether or not truly there will be actually really strong, stable structure, I'm not sure how that can really actually come out. But for example, non-coding RNA, certain non-coding RNA do have a really strong 3D structure, right? So I think it's one status, but you need to think about it as a sampling out, so you have a more diversity there. So I think rather than the protein, you just go with one structure. You should actually go with more. I don't think a cryo-EM only give you one structure. Actually, looking to cryo-EM, you actually have a sampling of a structure. It's just a lot of time, these structure data set was not actually taken out as taking count of that. So I think there's a lot of space on that.
Thank you for the beautiful talk. I really like it. For the foundation model part, for mRNA, can you think about something to design novel, better mRNAs like what I do, but you can probably do better. But on the other hand, I have a concern with foundation models because it's trained on existing data, whereas you want to go beyond existing data to design much more stable and efficient sequences, which probably is not covered in your distribution. So what do you think?
Yeah, currently it really depends how much you want to run on the fine-tuning step, right? So basically currently all the evolution related things will coming up, coordination, cooperation, like a base pairing distance. It's all about evolution constraint comes out, right? So we actually have a fine-tuning step, which we actually adding the in vivo structure as the fine-tuning step to actually make the prediction far more better. So that is really depends. You're using one data set for fine-tuning, or you're using a lot of data set for fine-tuning. So that's the next layer we want to go with. Yeah.
Thank you so much. I'm sorry, but we have to move on. But if you had a